So each person has three different events that result in genetic variability. The first of which is the independent assortment of the chromosomes, which we'll talk about next. Then we're going to talk about the crossover of the homologues or homologues, which occurs during the prophase 1. This was discussed also in chapter 27. And then finally, there is random fertilization of the eggs by the sperm. So, on our next slide, we're going to look at independent assortment. And during independent assortment, there's many different possible gametes that could actually result. So, in this case, what we're looking at is six different couples of chromosomes, which are going to end up forming various tetrads. So in this case, if we have 2 to the 6th, this means that there are going to be 64 possible gametes. So there's really an exponential increase in this concept of independent assortment. In the case of humans, though, since there are 23 chromosomes and pairs of those chromosomes, obviously, that means that there would be 8.5 million possible gametes. So, an incredible amount. So, this is a very simplistic example of the independent assortment. But you can also already see the different possibilities. So the green representing the maternal chromosomes, the purple color representing the paternal chromosomes, and we can see that all of these are possibly different. So it's pretty unbelievable. So the chance is going to determine how these tetrads are formed. So remember, the alleles are on different pairs of homologous chromosomes, and then they're sort of segregated independently of each other to different gametes. The second possibility, which is going to cause this genetic variability, the second source is what we've already learned about a little bit. So remember, genes on the same chromosome are linked, and it could be passed to the daughter cells as one unit depending on their specific location. Remember, the location is actually what's referred to as a locus. That is just the location of the allele on the chromosome. So during crossover, which is also called chiasma, the homologous chromosomes can break even between the linked genes, and there could be a precise exchange of these gene segments that result in recombinant chromosomes. So there, however, is a chance that the crossing over diminishes the closer that they are. So, for example, if this is one of the chromosomes, and we have one gene or one allele here, closer to the centrosome, and another one also, there is less chance of crossing over or chiasma actually occurring just because they're closer to each other. So the further away they are on the chromosome, the greater the chance that the crossing over could occur. So our next slide shows this crossing over process. And in this case here, we have genes that are for hair color, and we have a different gene that's for eye color. These are the homologous chromosomes, so these are the paternal chromosomes and the maternal chromosomes here. And you can see that the homologous chromosome synapse during the prophase, remember prophase 1 of meiosis 1, and then each chromosome consists of the two sister chromatids. So remember, this region here would be called a chromatid. So non-sister chromosomes, then they could essentially trade segments of DNA, and that is a source that increases genetic variability. So on the second step here, we see that one chromatin segment exchanges positions with a homologous chromatid segment, and so crossing over does occur to form this chiasma. So the point of crossing over is specifically happening right here. So what we're going to see on the next step is there's going to be an exchange between these different alleles, one for hair color, one for eye color. So we can see in number 3, the chromatids are broken at the chiasma, and the broken ends then join to their corresponding homologue. So we have a lot of randomization again. Here is a dominant allele for hair color. These are recessive alleles for hair color. This is a recessive allele for eye color and a dominant allele for eye color. So what we end up getting, again, is a lot of mixed up gametes. So, for example, in this first gamete, gamete number 1, we have blonde hair and blue eyes. And that's represented in gamete 1 by a lowercase h recessive and a lowercase e for recessive as well. The next gamete is also going to have blonde hair. However, they're going to have brown eyes. You can see that one of the alleles is recessive for hair, and the other allele then is dominant for eyes. So therefore they have brown eyes. The third possible gamete is going to have brown hair and blue eyes. So again, another possibility. There's a dominant allele for brown hair and also for a recessive allele for blue eyes. For the fourth gamete, there is an allele for brown hair and brown eyes. So both of the alleles are going to be dominant here for gamete 4. So when the chromatins segregate, each gamete will receive a unique combination of the parental gene. So that is yet the second factor, the second source of genetic variation.